And we come to the second speaker, which is Professor Broszka. Was it pronounced right?
Yeah, correct.
From the University of Warsaw. And he will talk about lab-on-a-chip technologies for testing drug sensitivity in 3D cell cultures. So a very important topic also because these are normally said to not only mimic tissues in a better way, but I'm not sure whether you will speak about tumor spheroids, which are also said to resemble cancer stem cells in a much better way. Okay, thank you very much for coming.
Thank you very much. Let me first thank Beat to accept my talk. And I would like to pick up you from the protein engineering to the classical engineering, which is focused on the design of a novel platform for the 3D culture, long-term 3D culture. Of course, in this audience, I'm not really able just to tell you all of the difference between in vitro and in vivo. I would like only to stress the dimension which is important if we like to use a lab-on-a-chip versus a Petri dish or the beaker scale, because in a beaker scale you have unlimited access of substrate. There is a nutrients access. It means otherwise the dimension of a beaker scale or a Petri dish do not let us to mimic the really in vivo situation. In case of a lab-on-a-chip, when we have a limited size of a microfluidic channel, all of these parameters like a substrate concentration profile and the nutrients concentration profile, as well the oxygen, is already quite limited, which is much close to the in vivo condition. Of course, there are other, let's say, advantages of this lab-on-a-chip devices. I do not want to stay too much time. Those who are interested, I'm open for any, let's say, the question and later on discussion in lobbies. What was our goal? Just to design the system novel platform to study to cell culture in three-dimensional. One of the most popular up to now, and it is, let's say, accepted as the best cancer tumor model delivered or, let's say, developed so far is a spheroids, which is multicellular. You can see here there is a necrosis core surrounding viable cells. Our first approach was to design a quite simple system based on the microfluidic, which contains here the channel. When we introduce the cell suspension, then part of the cell is seeding to this chamber. The size here you can see the microchannels is a 50 micrometers depth, 1 millimeter is wide, and this is cylindrical microwells is a depth of 100 micrometer. Idea is that this suspension cell, part of them is seated on the bottom, and then within 24 hours we can observe already spheroids create inside this chamber. Of course, to optimize all of the factor concerning diffusion, to have, let's say, the optimized condition for nutrient access, substrate access, we did the modeling based on the glucose solution to see whether there is optimized the depth of this chamber versus the diameter. And then we studied the long-term feeder culture in the cell using two different cell culture. This is a colon cancer cell. Another one was studied also the liver cancer cell. And we have seen that after, you can see after almost 20 days, we have the kind of situation which, of course, excuse me that I would like to say, this is like a homeostasis-like. It means that there is no proliferation and the high viability of a cell are still observed. Then the real challenge was not to do the, let's say, the setup which is available in our lab, but the idea is just to sell and thus to offer this setup to others to study and to be able to operate this setup. Then we said, okay, this is a cellular model which should mimicking at least in vivo. This should give reliable analytical signal, reliable value of a measurement, and then this should be acceptable by the through output, high through output, let's say, the principle. Then we decided to develop further on so-called the spherochip, where each chamber consists 18 small chambers to carry out the cell culturing of this spheroid. Idea was to combine, may I just further go on, to combine this, our spherochip, with a standard plate reader, which is used in almost all laboratories to use our system in combination with this already known system. And in each of these small chamber, we have one spheroid. Here you can see we change the shape of this small chamber from the flat one to the oval one to control and to have a more, let's say, a more repeatable size and the shape of these spheroids. This is just two examples, and this is let's say, the fluorescence photo of this all of spheroids, which you see. This is a one big spot containing 18 small chambers for the spheroid culture. This is an example. What we noticed already at the very beginning, that different cancer cell grows in different, let's say, in different rate. For example, the colon cell culture grows much smaller as a spheroid versus a liver cancer cell. Then the idea to combine, which I already mentioned, is just to measure fluorescence of this big chamber containing 18 small microchambers, which each of them contains one spheroid, to see whether there is a dependence of fluorescence intensity versus surface area of spheroids. And what we notice that there is directly, let's say, the proportional to spheroid size. It means also the number of cells inside these spheroids. A part of the staining of a spheroid, or especially the surroundings of a living cell, we also use another method to study the viability of this cell using the Alamar blue. In fact, we monitor metabolic activity of the cell. And this is done in this sense that we have a special, let's say, design microfluidic system which already generate us automatically 3 different concentration of a studying, let's say, the drug or any other reagent. It means that up to 7 different concentration we can design and we can automatically generate in the system based on laminar flow. So there is no any need to, let's say, to use any pipette or any system to generate different concentration. Then we can study several approaches which are very preliminary. First approach is that we have, for example, 5 selected this small chamber containing the spheroids. We add the drug. We test it based on the 5-fluorouracil, which is the most popular for the testing as a model compound anti-cancer drug. And then we add this fluorouracil with different concentration to each chamber. After several days, we let's say, control the metabolic activity of the spheroids as a surrounding cells. And then we can see that there is, of course, dependence of the viability of the cell versus the concentration of this drug. Then we did another experiment. We add 1 dosage of drug. Then after 8 days, we add once again the same dosage of this drug to see whether there is a difference between the multiply or 1 dosage the system. And this is a picture, a photograph of a fluorescent intensity of this smaller chamber. What is interesting that this platform is able to help us to control 3 different factors that we can use. We can study the multi-dose I mean, the administration of our cell. Then we can see also the resistivity versus drug of the different cells, because this is in time, and we do not staining the cell culturing. Cell culturing is permanently living, and we are just controlling, adding some agent from time to time, depends on our protocol. And the third one, which is maybe the most interesting, not for us, for our colleague from the hospital, that we can see that there is let's say, I don't know if I correctly translate, delay resistivity of a cell versus drug. So for example, here, you can see that after first dose, this is the same chamber, the same spheroids. After the second dose, we see that there is after first dose, viability of cell increase. And again, when we add the next dose, it already goes, let's say, in expected direction. So I think that we design and show, let's say, the some efforts which could be interested for audience, for all of others, you who are interested to collaborate. As I would like to stress, we are analysts, engineers delivering methodologies and tools, and would like to encourage others, doctors, physicians, and others for collaboration, because altogether could be done more than separately. Thank you very much for your attention.
Yeah, thank you very much for this nice presentation, a really useful tool. Other questions from the audience? Please. We have only one microphone runner, so sorry for that.
Hello, thank you much for a very interesting talk. I have several questions, but I keep it short. Can you control the cell number in the wells? So for example, when you start to load from the left side or the right side, and you have 18 wells or something like that, will the last wells also contain the same amount of cancer cells on the chip?
Here?
On the chip?
On the chip, this is a small chamber, contains only one spheroid. And the size of this spheroid is reproducible, is roughly 120, 130 micrometer. And we are controlling, I do not have a time to show other, let's say, our results, but we are controlling the size of these spheroids inside this microchamber.
Very nice.
So this is let's say, controllable.
Okay, and I'm pretty sure you have a coating in the well. Is it hydrophobic?
It's a hydrophobic, it's a PDMS.
Ah, okay.
PDMS just to guarantee that there is no adhesion of a cell. We also practice in the past adhesive cell, but this is a quite different story.
Okay. Then just short, and the channel between the wells was around the diameter was around 1 millimeter. Did I get it right? Between the wells, was it one millimeter? The channel to load the wells.
Well, okay, sorry. You mean about this principle?
Yeah. We can talk about this later.
Sorry.
Yeah, that is a good idea. So we have allowance for some more questions.
Thank you for a nice presentation. As you know, basically in the microenvironment, there are not only one type of cells. Tumor cells are the least usually cell population. So can you introduce other cells in this, and will it be affected by the flow in the chamber? Because different cells, I would assume, would flow differently, and then can you control the final composition of the tumor spheroids?
Thank you very much for this question. I expected I have 2 slides, but I would like to skip my, let's say, time to. We have another project which is designed just to study co-culture and mixed culture of different, the cell culturing. Also including the 2 parallel channel, which is connecting by the narrow gap of a channel, which led about the 30 micrometer, which led the cell to communicate, to contact, but do not mix. However, we can also some other, let's say, design to just to proceed the co-culture and the mixed culture. So there is only 15 minutes, sorry. But we are doing the same as well.
Good, we have allowance for one more short question.
What what I noticed that cells in your spheroids after several days become inhibited against division. So it's kind of contact inhibition. Normally, cancer cells are not inhibited. They can divide all the time. So could you explain why the cells are inhibited if they are cancer cells?
If you see as a spheroid, this is a necrotic core and this is viable cells. What we are doing, we are also washing from time to time the spheroid to remove the dead cell and remove those cells who are not attached to the spheroid.
So I see. So cells which are in suspension, they are removed. And
yeah, because what we like to study the spheroids as itself to control. Otherwise, we have, let's say, the not reproducible results, because in this case, we have in chamber one spheroid and the free cells. So to omit this, let's say, I don't know, the not controllable results, we'd like to control, to wash the chamber from time to time. Okay?
But it seems also to be the case that after prolonged cultivation, you have the entire well filled up with a large spheroid.
Yeah.
And that's maybe another kind of mechanical limiting factor. Then it may depend on when you apply the drug in order to direct which phase you would actually like to hit. Okay, thank you a lot again.